Thanks, Kevin. Thanks for the invitation and a very kind introduction. Feel like you summarize our lab's work in your own words. Nothing shows up in my website if you check. It's really Kevin's version. Really, yeah, really appreciate it. It's my great honor to present some of our recent work, kind of in between statistics and AI. I will cover three aspects today: on theory, on method, and on data. So before I go into these three topics, I want to briefly talk about my experience at UW. So you may find this funny. So about 9 years ago, I went to a summer school at UW held by the Biostat department. At that time, I was a second year math PhD student doing some crazy Riemannian geometry, useless stuff. Then I magically found this summer school. I found it very interesting. At that time, I have zero knowledge about Biostat. I had no idea. Then I just submitted my application because it's free, and I was admitted. And surprisingly, they covered the flight, the lodging, meal, everything. I was shocked. Okay, so I went to Seattle. I got this mark, which is really high quality. I'm still using this mark today. I had this for my morning coffee today, but because I'm not at home, I cannot show you the mark now. But really, thanks to UW, the summer school, I got my first exposure to Biostat, and eventually I joined the Biostat department for my career in 5 years. And by the way, I'm not sure whether the summer school is still running now, maybe not. So first, I'd like to briefly talk about the background. Mainly, I suppose people will agree with me. AI is very popular. As shown in this figure, this is from a Stanford annual AI report, where they summarize a lot of notable AI models. As you can see, each dot here is AI model. There are more and more methods in the past 2 or 3 years. This is also evidenced by the stock market. If you're interested, this is the NVIDIA stock market. You can see from the birthday of ChatGPT, about late November of 2022, the stock market was crazy. This is basically because nowadays, if you want to do AI research, almost surely you need to either buy some GPUs or you want to run some GPUs for your research. In our statistics community, there are some recent discussions about stats and AI. For example, there's a recent Harvard Data Science Review paper discussing stats and AI. In the conference, for example, JSM last year in Nashville, the topic was related to AI. And for the coming INAR conference in March, the topic is also kind of stats in the AI augmented world. So talking about the stats journals, for example, JASA has this recent special issue on stats and AI, and Annals of Applied Statistics also have a special issue. The deadline, I believe, was last December. It's also about like stats and AI. From the funding agencies, the National Science Foundation, ASF, has some special calls on AI and math, AI and biomedical studies. Similarly for NIH, there's a so-called Bridge to AI Consortium, which is really designed to, encourage biomedical research with AI collaboration. And so let's be more concrete. If you look at the Annals of Applied Stats website, look at their special issue, they are particularly interested in 2 types of work. One is how statistics helps AI. This happens to be my first topic today. And the second type of paper they are interested in is in the other direction, how can AI help statistics? That happens to be my second topic. And this is a snapshot from the NIH website. They listed about 5 types of work they're interested in. The first type is we want to generate biomedical data that are ready to use for AI researchers. This happens to be my third topic today. Okay, so let's move on to the first topic, which is more on a theoretical aspect of how statistics can help AI. AI is a big topic. Let's narrow it down to be more concrete. The type of AI I want to discuss today is called the generative AI. It's not because it's easy to analyze, it's really because it's a very important hot topic. There are a lot of exciting products in this field. For example, ChatGPT can generate text. This OpenAI DALL-E 3 can generate images or generate music, video. AlphaFold can generate new proteins, or Copilot can generate computing code. I suppose students may have been using these tools. For example, some of my students use Copilot to write code. It's much more efficient. They use ChatGPT sometimes to polish their papers, their report, maybe just to write their paper. There's a lot of debates in the community about using ChatGPT. For example, some guy submitted like a 40 NIH proposals using ChatGPT. That's why there's a recent constraint, each PI can submit up to, I don't know, 6, something like number of PI grants every year. And so for illustration purpose, let me show you a very simple example about how this generative AI works. So usually we start with training data sets, like here, we have a lot of dog images. Then we throw this into some AI model. At the end, the goal is to generate new data that look like real data. For example, this generate a real dog image. This is my favorite example. So that's basically the rough idea of generative AI. You hope to generate new data that are kind of real. Among these generative AI, I am focusing on a special type of model called decoder-based model. Again, it's not because it's easy to analyze, it's because it's the most popular or most commonly used model in practice with a really good performance in practice. So here's a pipeline. We start with a training data set. We throw this into usually a black box model. You spit out a function G. I call this generator. That's why I use letter G. And with this G, we can do data generating. For example, you can choose a simple latent space. Here I show a 3-dimensional cube. That's easy. I also choose a latent distribution. For example, I do a uniform distribution. It's probably the most simple example here. Then I sample random variable Z from this uniform distribution. I just apply the function G to this Z. And the resulting random variable GZ is exactly what I want. It's just an image I want to generate. And so the reason to study this is because this decoder-based approach include a lot of popular models. I mean, you may have heard about this. For example, variational autoencoders, generative adversarial neural nets, diffusion models. If you're interested in large language models, this covers GPT families, Llama, Mistral, etc. So they are all decoder-based, or they involve some decoder in the model. And to understand or to study this theoretical aspect of the model, I hope to translate it to a statistical problem. So do some translation here. For the training data, we assume there's a true underlying data distribution Q, and my observations are ILD samples X. Then through this black box, I have a generator G, nothing different. And I have a base matter rho in a simple space, and I sample this Z from the base matter, say uniform. Then this GZ, the image of interest, follows the push-forward matter of rho through this generator G. And apparently, this GZ is the estimator of the true data distribution Q, denoted by Q hat. And hopefully, this generating distribution Q hat is close to the ground truth Q. And you may find this problem familiar. This is actually sometimes called density estimation problem in statistics. For example, if you do a simple Google search, you can find some textbooks. And the list is the year when they were published. And apparently, this is not a new topic. But if you really read the theory in these papers, they may propose a concept called the curse of dimensionality, which means that if your data is really high dimensional, the performance will be really bad. But the practical observation is a little bit different in the sense that even if we deal with high dimensional data, say high resolution images, where the dimension is the number of pixels, it still works really well for those deep generative models. We want to understand why it works from the theoretical point of view. So a common belief is that high-dimensional data are usually in the low-dimensional space or low-dimensional manifold. This is the so-called manifold hypothesis. And motivated by this hypothesis, there are some existing theoretical study in this direction. For example, there is a very beautiful paper by a group of people at Georgia Tech Math Department published on NeurIPS 2022, which studied this approximation power of deep neural networks or deep generative model. Here is a brief recap. So assume the true data distribution Q is supported on some remaining manifold M with a much lower dimension, small d, than the data dimension, capital D. Then there exists a constant alpha so that for any approximation error epsilon, there exists a deep neural network G in some function class I will define in a second, such that the Wasserstein-1 distance between the push-forward matter and the true distribution is arbitrarily small. So this Y is called approximation power. It basically says that my deep neural network G can learn any distribution through the simple push forward. where this G is a function from a low-dimensional space, D plus 1, to the original data observation distribution, or a space in dimension capital D. Moreover, there are no more than L layers in the neural network, no more than N neurons in the network. And here, rho is an easy-to-sample distribution on the simple unit cube. For example, it can be truncated Gaussian, it can be uniform, it's up to your choice. So then this function class just represent all functions or all deep neural networks with no more than L layers and neurons, and the input dimension is D plus 1, output dimension is capital D. That's the notation. Moreover, they can control the growth of number of layers in neurons, which is also called the complexity of the network, where the number of layers grows in a log rate, and the number of neurons grows with epsilon in a polynomial rate, where the rate is D over alpha. And the key here is that the data dimension D is not even in the result. Instead, it's the intrinsic dimension, which is standard observation in manifold learning literature. This is kind of not surprising. You may also wonder, okay, this is just a theoretical guarantee. There exists such a neural network, but in practice, how can I find it? They also have a statistical guarantee, which is more like a practical version. Instead, we have IID samples x1 up to xn. Then they show that there exists some empirical risk minimizer. You can imagine this GN hat is some output from a deep neural network model, for example, VAE or GAN. This is something you can really calculate in practice. Then the Wasserstein 1 distance is also arbitrarily small, where the rate is 1 over D. This delta is some technical constant. You can treat it as 0 in a non-rigorous way. And here again, G is a deep neural network where we can control the number of layers of neurons in terms of the rate of the sample size. And then with this very beautiful result, we can ask two immediate questions. Question one is, we assume the data dimension, intrinsic dimension is D, but why the input dimension must be D plus 1? This is like mathematically it's not that elegant, or maybe you can argue with okay, I don't care whether it's D or D plus 1, I'm okay with the additional dimension. But there is even a more important question here is that in practice, we do not know the true dimension of the underlying manifold. Instead, we just observe high-dimensional data. And if you look at the literature, sometimes you can argue you can estimate this using the data, but actually estimating D itself is a challenging problem. It's probably as difficult as estimating the distribution Q. And in practice, people do not really estimate this D. If you look at the packages, they sometimes treat this D as a tuning parameter, or sometimes they just do some hand-waving choice. For example, 256 or 512 due to some hardware constraint to be convenient. And the question is, why does it still work, even if you do a hand-waving choice of the dimension? Because almost surely your dimension you chose might be wrong, right? It can be lower, it can be higher than the ground truth. A more general question is, how does this input dimension affect the theory or the asymptotic behavior of this generative model? So to answer the question, we developed some recent theory just designed for this case. First, we will show the approximation power as the 2022 paper, where we show that under the same assumption as before, nothing new, for any input dimension m between 1 and the true data dimension, we can show the same result. For any approximation epsilon, we can find the deep neural network G so that the distance is smaller than epsilon. And the main difference here is that in the previous result, this m must be D plus 1, one higher than the true dimension. But here we allow this m to be anything. Again, G is the deep neural network, but the input dimension is m. It's like a non-trivial extension. Moreover, we can show the number of layers still grows in log rate, but the number of neurons has a very interesting behavior. If the manifold dimension is underestimated or your input dimension is high enough, this becomes the same as the previous paper. But if you underestimate the manifold, if you are lazy, you want to use a small input dimension, there's a very different rate. Here is the input dimension divided by some alpha, but this alpha is not a constant anymore. Instead, it depends on both the input dimension and the approximation epsilon. I will spend more time on this. That's the most interesting part. So I just circle it here. And then I talk to my student, okay, there's no free lunch. If your input dimension is low, you must pay something. Otherwise, you should always use like one-dimensional input, right? So the price might be, if your approximation error epsilon is smaller, your alpha is also smaller. That means your final rate is much bigger. And eventually I had a conjecture that the alpha term will go to zero as the approximation error goes to zero in this case. This will lead to a super exponential growth. It's like infinity to infinity. It's a crazy growth. And to also study the statistical version, we also have the analog using a finite sample. And again, there's the empirical minimizer GN hat. We have the same results as before. And again, if you underestimate the manifold dimension, there's a different rate. And again, you can show that this denominator alpha goes to zero, where you have a super exponential growth. That's kind of the price you pay if you underestimate the manifold dimension. And you may ask, why it really works. For example, our results suggest that any m you will make it work. For example, if m is equal to 1. And I learned from basic topology that a continuous function cannot increase dimension of an object. For example, here, if the input space dimension is 1, your input is the unit interval. The deep neural network is a continuous function. It maps in unit interval to a curve or a smooth curve. And the smooth curve is still one-dimensional. Why it can understand high-dimensional distribution? My student, Kevin, who led this project, gave this very nice intuition, is that look at this space filling curve, which is a common tool in topology. So we start with a simple curve, and we make it more and more complicated. Eventually, this curve fills the entire space, which is two-dimensional. This is an example where one-dimensional object can fill two-dimensional space. You can extend this to a higher dimensional case. And the main challenge of this proof is that in the standard case, say in 2022 paper, your target is the distribution Q on a fixed manifold M. But in this underestimated case, when we make this epsilon smaller, the space filling curve is changing. And actually it's getting nastier and nastier. It's a crazy curve at the end. Then you want to learn a distribution Q epsilon supported on a crazy space M epsilon, which is changing over the approximation error. So that means all the constants in the previous proof are not constant anymore. So you need to trace all the constants. For example, if you have some theoretical work before, if I ask you to track all the constants, you'll probably kill me. It's very annoying. And so what's more challenging is if you read this 2022 paper in the proofs, they didn't discuss the constant. So we have to go back to the reference of the 2022 paper, which is this optimal transport book. This is like the Bible in this field written by Villani. And if you work on, versus and distance, it's not a bad idea to read the book. So I believe I can find the solution in the book. So I spent some money on this. It's still in my office. I'm not sure whether you can see it. It's very thick with about 1,000 pages. When I bought this book, I spent $80. I tried to find the proof. It didn't work because even in this book, they didn't discuss the constant. But fortunately, I found that in this book, after each section, Villani wrote a very comprehensive literature review. Then he pointed us to even more earlier citations that study the constant. Eventually, we were directed to a series of work by a mathematician, Louis Caffarelli from UT Austin, who studied this partial differential equation called Monge-Ampere equation. And using the tools in this series of papers, we finally managed to show this alpha goes to zero, which conclude the super exponential growth. However, we didn't really solve the problem completely. For example, if you set this approximation error to be fixed, so for finite epsilon, you may ask, what's the corresponding alpha? This is important because this alpha determines the converse rate. But unfortunately, we do not know how to calculate this alpha. Actually, even in the 2022 paper, this alpha was just proved to be, exist. They didn't really show how to calculate in practice. So I would say this problem is not fully solved. You may also wonder, okay, this is just our proof technique. Is this really the case in practice? So motivated by this, we did a very simple simulation. There we have a two-dimensional uniform distribution at the target, and we use a generated adversarial neural net. You can see that as we increase the number of iteration, the orange points are the generated points. It start to fill the space, especially if you look at the middle one, at the iteration of 1,000. It's kind of really filling the space. And if you look at the training loss on the top right, That's standard. But if you look at the top, bottom right, is the space filling distance, which is the distance between the curve and this two-dimensional square. You can see it's highly correlated with the training loss in this generative model. This suggests that actually the model is learning the space filling curve without really aware of it. So quick recap, we discussed the generative models where we show that any input dimension works. It kind of coincides with the observation in practice that randomly choose a dimension may work in practice. But we also show that there's no free lunch. For example, if you really want a low approximation error and you want a low network complexity, then you have to have a high input dimension. You have to sacrifice one of the three vertices in the triangle, and we call this impossible triangle. This also coincides with practical observation in the sense that, okay, if your input dimension is pretty low, you sometimes need a very big neural network. And there are some interesting future work. For example, there's a phase transition in the number of neurons. It can be polynomial or super exponential. Can we use this phase transition to estimate the true dimension of the manifold? Secondly, can we use this theoretical result to guide method development? So far, it's purely theoretical. We don't know how to use it in practice. But I'm sure, with this theoretical insight, we can improve or develop a new method in this deep generative model framework. So that's the first project I want to discuss. Then I will move on to a very recent work. Actually, I was just talking to Kevin. It's not even in my abstract. The paper is not finished yet. It's not even archived, but we have some exciting result I want to share with you. So this problem is known as recursive training. Imagine that OpenAI collected some real data from the entire internet. I call this X0, which is sampled from a true data distribution P0. And OpenAI used this data to train their GPT 3.5, and people like it. And then people use this GPT 3.5 to generate new data through this learned distribution P0 hat. And we call this generated data Y1. At the same time, OpenAI collected some new data, X1, from the real distribution, and then they combine X1, Y1, essentially because the first model works really well. It's hard to tell whether the data is real or generated. So in the training data of GPT-4, there might be some real data or might be some fake data from the previous model. They use this combined data to train their GPT-4, the learned density is a P1 hat. And you use this to generate another data set because people like it, and you have another collected real data, you get another estimator. You keep doing this. And the question here is, does the learned distribution PT hat go to the ground truth? It's like a consistency question as the number of iteration T goes to 0. So you have GPT infinity. Does it really work? And this problem is known as recursive training, which is arguably a pretty important topic nowadays. But surprisingly, the existing work is pretty sparse. For example, there's a Nature paper in 2024 whose study is called AI model collapse. But surprisingly, the assumption is very strong. They assume the true data distribution is Gaussian, and their estimator in each iteration is also Gaussian. Then they can show that the learned distribution PT hat is not really converging. It's far from converging to the ground truth. Instead, they show the distance actually blows up. It goes to infinity. Also, the covariance almost surely goes to 0, meaning that you go to a point mass. That's why they call this model collapse. It's more like a negative result. Later on, in the same year, there's another paper studying this problem in a similar setup, but it's regression. You assume your predictor follows Gaussian, you have some noise, and your response variable y is a linear model of the input x with some ground truth regression coefficient beta 0. They show that under this recursive training, the distance between the estimated regression coefficient is not far from the ground truth. That means it doesn't blow up, but it doesn't show it's consistent. It just shows the distance is bounded. It may go to 0 or not. So this is kind of surprising in two ways. First, no one really showed the positive result, like consistency. Second, they also have this very strong Gaussian assumption, which is not really realistic, we argue, because in practice, the real data is complicated, and those deep AI models are not Gaussian, right? So to answer this question or to address the gap, we kill this Gaussian assumption completely. Instead, we assume there is a base model, And the base model can be anything as long as it's consistent in the sense that if you have infinite data, your learned density will go to the ground truth in a polynomial rate. And the rate can be anything. And this pretty much cover any density estimation we learned previously. For example, in stats models, there's a so-called kernel density estimation or generative adversarial neural nets or diffusion model. They all satisfy this consistency assumption, but the rate might be different. And the second assumption is also pretty weak. We assume the distance considered here is convex, which include total variation, Wasserstein distance, MMD. This is also a broad type of distance matrix. And then we assume there's a real data fraction in each iteration, alpha between 0 and 1. And under this very mild assumption, we show that the estimated density after iteration t is actually consistent. And what's more interesting is the convergence rate is the minimum of the base model rate p and the real data fraction alpha. Let me spend more time on this. Okay, so if you work on the first row, you have a base model, and the best rate you can achieve is a half. Say the best p is a half. This result means that as long as you have half data to be real, your final convergence rate will be a half. That's optimal. But if your real data fraction is below 50%, then the resulting rate will be worse than the base model rate. So this is alpha. For example, if you have a 20% real data in each iteration, your rate will be 1/5 instead of 1/2. But if your real data fraction is 0.8, it's the same as the case where it's like 0.5. So there is a very interesting phase transition. We have some simulation, but due to the time limit, I'll skip it. But instead, let me show you an interesting example, which is a real one we can visualize. We consider the base model to be a diffusion model where the rate is arguably close to a half. And we consider a case where we have only 30% of the real data. And we will show that the model still works. So here is our training data, some MNIST images. We call this X0. You throw this into a diffusion model, you get some generated data, which is really bad. Then we combine 70% of this fake data and 30% of the real data, X1, Y1. We throw this into diffusion model again. It doesn't really improve. You keep doing this after 5 iterations, you can see it's still bad. The generated data is still bad. But if after 50 iterations, you can see the generated data much better. There are some images that look real, but there are some still like a blurring, purely noise. And 200, it's much, much better. And a few moments later, you get some reasonably looking images. So this is just an illustration. It shows that even if you have this recursive training with a fraction of fake data, we can still guarantee consistency. This is really challenging the previous result about the model collapse. The conjecture, the reason is previous models assume Gaussian, which is pretty restrictive, but we don't assume any parametric assumption or parametric form of the true distribution. We don't assume any parametric form of the base model. So that's probably the flexibility in the true distribution and the model distribution make it work. A quick recap. We discussed this recursive training issue, which is increasingly important in AI or generative AI. We show that if you have a mixed data of real and synthetic, you can still get consistency. More interestingly, the rate is determined by both your base model rate and your real data fraction. And there's an interesting phase transition. Some future work. Here, we assume the sample size in each iteration is fixed, and the fraction of real data is also fixed. What about the sample size is varying? Maybe OpenAI has more data in the next version, or because eventually there will be more and more fake data on the internet, the real data fraction alpha t may decay with t. In this case, how does this change the theory as an interesting future work? So that's all I want to talk about for the first part, which is more like the theoretical aspect. Then let's move on to the second topic. As I promised, this is the second type of paper or work AI special issue is interested in, that is how to use AI to help with statistics. Let's look at a concrete example, finally a bio-related example. So we have 2 cancer cell lines where we collect the expression of over 700 genes. And the scientific question is very simple. Do these two cancer cell lines have the same gene expression on average? And you may translate it to a hypothesis testing problem. This is led by my former postdoc, Jordan Bryant, who's now a faculty at UVA School of Data Science. So here we assume the gene expression follows a Gaussian distribution with known covariance. Bear with me for a moment. I know it's simple. And with potentially different mu vector, mu1, mu2. And the scientific question can be translated to this hypothesis testing problem, where the null hypothesis is they have the same gene expression versus they have different gene expression. And if you read your stats 101 textbook, this is trivial. I know how to do it. I can calculate this quadratic function, I get the t-statistics, I do a t-test. So my postdoc asked this very interesting question is, can we improve this? So what's the intuition or what's the motivation to improve this? Is it really possible to improve this? And here is the observation. We have about 700 genes, and we know these genes are not independent. Some genes are known to be positively correlated. Some genes are known to be negatively correlated. So mathematically, it means we believe the difference of the mean gene expression vector lies in some low-dimensional subspace, which captures the correlation between genes. And so some immediate questions follow. Is this really true? It must be true, otherwise I won't present it. So if it's true, how can we find such a subspace? If we can find such a subspace, how can we use the subspace in our hypothesis testing problem? So we want to answer these three questions one by one. So let's step back a little bit, look at the data. This is the data set we got at the beginning from our collaborator. I call this biologist view. Each row is a cell, each column is a gene. Okay, I send this table or this CSV file to my student. Hey, Kevin, can you help me to do a t-test? My student translated this data matrix to the second one on the right. I call this a statistical view. This is what we learned in Stats 101. We try to be general so that we can apply these tests to all applications, not only just gene expression. But the issue is during this translation, we kind of kill the information in the column name or the gene name. For example, if you talk to your collaborator, they will tell you a lot about this TP53. But if you ask them about feature P, they have no idea. It's a general column name. And so the idea is, can we bring this information back, which is lost in this translation? So what do we know about these features or these genes in this case? Unfortunately, there's a website called GeneCard. You can just type a gene name, say TP53, it will immediately tell you this is a, this encode a tumor suppressor protein, blah, blah, blah. It's very organized, principled, and actually every single sentence here has been validated by wet labs. The scientific researchers and scholars spend a lot of time and effort making this website. It's really well organized. Why not just use it? It's a free resource. And so the question is, how can we use this information? And obviously, large language models are really good at understanding language or text. So there's a recent encouraging paper by Chen and Zhou published in 2024. And I think Chen was a PhD student in your department, graduated a few years ago, where they throw this text description of this gene card into a GPT embedding model, and then they get a vector representation of this text. It's like a numerical representation of this biology. And they show that they can use this embedding for some downstream tasks like clustering, phenotype prediction, something like that. This is pretty encouraging. It means that this black box embedding vector, which is numeric, can kind of represent some biological information in the text. That's the motivation. Go back to our hypothesis testing problem. This is our assumption. This is our belief. And then from this embedding of this large model on the gene card description, we get this embedding matrix E. Each row is a gene. Each column is just a magic feature from this LLM. We have zero control on that. The 1536 dimension is just set by OpenAI. We cannot change it. Okay. Then we calculate the top D singular vector space. We call this ED. And we claim that my difference of gene expression is close to this ED, which is a crazy claim at the first glance, where this D is some low dimension compared to the number of genes, which is 700. This is not really belonging to it. I would say it's lies in or lies is close by. So how to validate this conjecture, we can do some real data analysis. Just taking this two cancer cell line example, we can project the difference gene expression to this singular space. We look at the explained variance, the higher the better, and we vary the dimension from 1 to 200. On the red panel, we can see the blue curve is the explained variance when D varies from 1 to 200. And the gray curves are random projections. The red dot is 95% confidence interval. The red curve is the mean. This figure shows that the embedding space or the singular vector space from this LM embedding on gene card is better than random. So what's that gap? We conjecture this is some true biological signal in the background knowledge in gene card. That's how we summarize existing background knowledge. So this is how we find the subspace. Very simple, just a singular vector of the embedding matrix. And the remaining question, which is the most challenging and important one, is how can we use this ED, the embedding, in our hypothesis testing? Again, this is our hypothesis testing problem. And because from this LM embedding, we can summarize existing background knowledge in a mathematical way, we can treat it as prior. So we believe the difference between this gene expression vector follows a Gaussian, where the covariance decomposed into two terms. The first term says the difference of the vector lies in or close to this low-dimensional subspace spanned by the columns of ED. And the second term is just non-informative. And then that's interesting, you have gene card website, you use it as a prior information through LM embedding. But remember that our original problem is frequentist. We have a hypothesis testing problem, and the prior is usually used in Bayesian statistics. And they sometimes conflict to each other. How can we integrate these two ideas together? Fortunately, there's a recent technology by Peter Hoff from Duke, which is called frequentist and Bayesian framework, or FAB. Using his probability tricks, we can show that we construct a new statistics T-FAB in this a little bit longer equation. This gives us a valid t-test. By valid, I mean we can guarantee type error control regardless of the embedding E. However, we expect that if your embedding E captures some real biology information, then the power of this new t-test or t-FAB is higher than the standard t-test. And just recall this t-statistics for the standard test, and the difference is as follows. If you let this gamma go to infinity, that means my prior is non-informative. I do not trust my large language model embedding at all, then my t-FAB correlates to the standard t-test. But in practice, if you trust your LM embedding somehow, or you trust your gene card information somehow, you can use a finite nu and finite gamma that will give you a valid t-test, maybe with a better power. And in the paper, we have some numerical evidence on real cancer genomic data where we can show we can increase the power a lot. You may also wonder, in the entire discussion here, I assume the variance or the covariance is known. And in practice, we do not know it. Actually, we allow for unknown covariance, but the statistics is nasty, so I won't show it here due to the time limit. And we also have extensions to other tests. If you're not interested in t-test, we can do f-test or something else. Then we can do one step further. What about we look at DNA level instead of RNA level? For example, you can do genetic variants, you can do variant level embedding and FAB test. I told my student, let's do this as another example is a low-hanging fruit, but actually I didn't really see the snake there. So for the gene card example, it works really smooth because we have this gene card website. We can just use it in a free way. But for the genetic variant embedding, there are a few challenges. First, as far as we can tell, there is no known standard description of the genetic variant. Second, the scale is also different. For the gene PD embedding, there are about 30,000 genes. For genetic embedding, there are millions of SNPs in UK Biobank. If you look at the whole human genome, there are about 9 billion genetic variants, and not at the same scale. Also, sharing this genotype data is also kind of pretty complicated than sharing gene expressions. To overcome this, we first do the annotation. where we got 3 data resources called Favor, GWAS Tag Catalog, and Cleanvar, which captures some existing knowledge about the genetic variants. We hired a smart research assistant, this monkey, also my PhD student Hong, and we have support from industry partners. Eventually, we got this annotation of all 9 billion genetic variants in human genome. The second step is once you have the annotation, you can throw this into some large models where we consider 2 models, OpenAI embedding, which is not free. We have a QN3 embedding, which is free. Focusing on the UK Biobank level, we have 1.5 million SNPs, and we have different embedding dimensions depending on the model. For this OpenAI embedding, it's 50,000. It can be lower and can be higher depending on your model. Then you can do fab tests. It's nothing different as before, the same pipeline. And more interestingly is we can do individual level embedding in addition to the genetic variant embedding or gene embedding. Here we have this genotype matrix. Each row is an individual, each column is a SNP. You multiply this by the embedding matrix, SNP embedding matrix, you get the individual embedding matrix. Each row is an individual, each column is a black box feature from this LM. Then we claim we can use this P as a new representation of the genotype matrix, but we reduce the dimension from 1 million to 1,000. It's a big improvement. We show that we can use this P for phenotype prediction with comparable performance than using the raw genotype matrix. And we also argue there's little privacy issue because sharing this magic embedding vector might be okay because you cannot trace back to the genotype information. To summarize, we discussed how to use LM embeddings for hypothesis testing. Embedding is really all you need. We discussed 2 versions, genes and genetic variants. For the genetic variants, we have 3 different versions. One is UK Biobank with 1.5 million SNPs. Another is UK Biobank imputed with 90 million. The other is a human genome 9 billion. For the first 2 versions, we used OpenAI embedding where we pay about $1,000. We have a grant from OpenAI. And for the whole human genome, we have about 9 billion embeddings. The estimated cost is about 100,000. I submitted the grant, but OpenAI doesn't like the idea because they believe most interesting information is in the first two sets. For other genetic variants, we have really little understanding to them, like their association with disease or phenotype in general is really poorly studied. So we have to use a free model, which is QN3, is one of the best open source model. But another challenge is we need a lot of GPUs. So to run this embedding for the 9 billion whole human genome, we got some support from NVIDIA. We spent about 5,000 GPU hours. Finally, we got the embedding matrix, which is about 30 terabytes. We have the storage issue. Unfortunately, we got a grant from Hugging Face, which is very generous. We can upload our embedding data there for free forever. They promise forever at least. So for the remaining of the time, I'll quickly talk about the third topic, which is about to build data set or biomedical data set for AI researchers. This is motivated by multimodal AI. At the beginning, ChatGPT can allow you to input text, but after a year, it can allow you to upload a PDF, maybe a figure, or you can even talk to this GPT. This is known as multimodal AI, where you have different modalities of the data. This is also everywhere. For example, in EHR data, you have different modalities. And in the health domain, Google has 2 products. You can upload an X-ray, you can ask a question, or you can take a screenshot of your skin, you can ask similar questions. Another interesting data type we are interested in my lab is spatial transcriptomics, as Kevin introduced. So the data set is very interesting in the sense that it has different modalities by itself. For example, you have a H&E staining image, and you have a 2-dimensional spatial location, which spot. Also, you know the gene expression profile at each spot, which is really high-dimensional. So it has 3 modalities. Gene expression make genomic people happy. Spatial statistics people like spatial location. Computer vision people like this histopathology image. And this technology is called the Nature Method of the Year in 2020. And the proteomics version is called the Method of the Year in 2024. And essentially, it's a perfect type of data to train multimodal AI. However, for the existing ST data, there are some limitations. For example, if you look at these 2 very influential work, one proposed 23 slides for a human breast tumor, The other one proposed 12 slides from human cortex, human brain slides. But the number is still very small. It's because it's very expensive to get such a data set. Each slide may cost you a few $1,000s. But if you look at these notable AI models, the training data size in number of tokens is at the scale of like a trillion. So we really need a big data for this. So motivated by this, we need some data with images, with spatial omics, and with a large scale so that we can train this multimodal AI. Motivated by this need, my recent graduate student, Jiawen, who's now a postdoc at Stanford, curated one of the first and largest public available data set called ST-Image 1K4M. It captures over 1,100 slides, covers over 4 million spots. Each spot is an image and gene expression pair. Since it was released in late 2024, it has been downloaded by over 200,000 points on times on Hugging Face as of November last year. And this is just a quick summary. This dataset captures about 50 tissue types across 8 species, about 600 are for cancer studies. The 2 leading tissue types are brain and breast, and this work is supported by NVIDIA. What makes us more exciting is after it was released, within a year, we can find 6 foundation models trained using our dataset for, say, representation learning, for denoising gene expression imputation, or for zero-shot gene expression prediction. And so for the time limit, I'll skip some opportunities. I want to briefly summarize the work. First, I'd like to thank my collaborator Yixin from Michigan on the first project, my colleague Xihao on the genetic embedding, and my colleague Yun for the ST-Image dataset. And you can find the genetic embedding datasets through this first QR code, Hugging Face. Excuse me. You can find the ST-Image datasets through the second QR code, also on Hugging Face. Everything is publicly available, free to download. And this work is supported by federal agencies and some industry partners. I also like to thank my students. That's all for my talk. Thank you.
Thank you. That was absolutely wonderful. If people have questions, you can feel free to raise your hand, or if you feel opportunistic, you could just directly unmute if you don't see anyone else having a question. Or maybe I can start with a random question. So, I was super curious, well, about many parts, but including the first part. So, in your theorem or other theorems that you cited, the one that you drew a lot of inspiration from, how important do you need to get exactly the empirical risk minimizer? So I'm wondering how this relates to like double descent or like if we don't minimize it exactly, is it just a proof inconvenience? We still get those properties, or is the proof somehow, depending on how it's proved, do you really need specifically the empirical risk minimizer and anything a little bit off is, near worthless?
Yeah, it's a great question. I'll say it's kind of restrictive in the sense that you cannot use arbitrary estimator. But it's also kind of general in the sense that you don't have to use a really special model. It means that you need a reasonable deep generative model, but you really need a good risk minimizer.
I
If you
see.
do some tricks in your training, there's no guarantee on your estimator in theory. But actually, in our simulation study, we didn't literally use that risk minimizer because we just directly call the GAN package, or Wasserstein GAN package. They did something fancy in the package. I don't think it's exactly the minimizer. But it still works in practice. Sorry, I mean this one. So in theory, I don't know, but practically, I think it works.
I see. And is it specific about what the loss function has to be, or can I add all my like loss terms to encourage some special properties, but do those special properties all of a sudden
Yeah,
destroy
great
the
question. Great question. My conjecture is yes, but we didn't prove it.
I see, I see, I see, I see. Okay. Wow.
Hi, Didong. Maybe a follow-up question to that one. So, to what extent does the manifold structure come in here? Is it really is it just intuition? So could it just be a lower dimensional set on this?
Yeah, that's a great question. Actually, this goes back to the original question, whether the D plus one, actually, that's
Yeah,
related
sorry, I
to,
missed the first 10 minutes, by the way. So maybe
oh,
the
it's
answer was
okay.
there.
It is actually related to the 2022 paper. What they did is, because of manifold structure, you can look at a local neighborhood where you have a local coordinate. The local coordinate is d-dimensional. Then you can do
Okay.
some tricky math, some like a nasty calculation. Then you can show this locally. What they did is they use another dimension to paste the local patches together to make it a global claim. That's why they have this plus one. This one additional dimension is just for pasting patches together. That's why we believe you can improve the theory.
Got it.
Yes, to long story short, they've used this manifold local property. Locally is
So tangent
Euclidean.
space expansion, I guess.
Exactly, to
Yeah.
do the, yes.
Okay.
Thanks.
Maybe given that I made different questions on different parts, was the when you compute these embeddings for the 300,000 people in the UK biobank,
Mhm.
you have a 1,000 dimensional embedding, I think, a bit more. To what is the dimension of that embedding that you'd get you worried about privacy issues? I mean, of course, if you increase the dimension of the embedding, at some point there could be privacy concerns. Do you have a sense of what the dimension is and when we should
Yeah,
start worrying
that's a
about
great
it?
question. We didn't study this, but I believe there are some like a random matrices theory. You can kind of quantify how much information you can recover backward. But another kind of difference I want to highlight here is that if you use the OpenAI embedding, it's just black box. You never touch the embedding function. I would say it's extra safe in that sense.
Got it.
But
Yeah,
yeah,
that
so
makes
the quick
sense.
question is, I don't know.
Yeah, makes sense.
Interesting.
Thanks, though. Great talk.
Thanks.
Anyone else have a question, or I can ask another random one if no one else has one. I'll ask a more biology question just for the spice of variety. So for your gene card, this was super fascinating. Um, do I maybe there's already an option to do this, but when you're training, or I guess you're not training, you're just you're asking, you're querying the embeddings.
Yeah.
When you're querying these embeddings, did you need to additionally prompt what biological cancer system you were studying, or you put in some donor covariates because these genes might have different functions in different cells, different contexts. They interact with different cytokines, and or did you find just like blankly telling them I'm interested in these genes and without much biological
Great
context,
question.
that was
Great
already
question.
good enough?
Yeah, thanks. This is a very good question. This is kind of a confusing piece. I got similar questions before. I want to clarify that. I forgot this actually. So the GPT embedding model here is not ChatGPT. You cannot chat with the model. It's just a black box function. Your input is just a paragraph. You get a vector.
Oh,
It's not
oh.
random. You throw the same text, you get the same embedding all the time. There's no hallucination. There's no worry at all. You cannot prompt anything. So that's why it's kind of misleading. I call this embedding model. I didn't call this ChatGPT.
I
But
see.
related to your question, what you can do is you can design your description for different target. Maybe these are like generic information I'm not that interested in. You can design your annotation just for your, say, if I'm interested in a disease, you can only talk about the information about that disease. And we actually have some ongoing collaboration um, in some like a psychiatry world, and they are defining their annotation by themselves. They will write down what they are interested in, and we can just use LLM to summarize their background knowledge in a mathematical way. And actually, that's why I claim
I see.
embedding is all you need, or really annotation is all you need. Annotation, I argue, is the most important and first step. You need to collaborate with a domain expert to create the most meaningful embedding. Oh, sorry, to create the most important annotation. And the embedding is straightforward. Just throw this into existing model. You cannot even train it. And all the pipelines, it just follows straightforwardly. So all you need is to do a really high
See.
quality annotation.
I see. I see. And then this annotation typically is in your gene case. a little annotation for all 33,000 genes,
Yes,
and then
yes.
you,
For example, if you are not interested in genes, you deal with proteins. You need to
mm,
look at the description of each proteins.
I see.
So as long as you have some feature of interest, you want to give some background knowledge of those features. It can be like outside of biology even. And this LLM is just a principled way to make your background knowledge to numbers so that you
I
can
see.
use it in productive tasks. So we argue
Oh,
it's a pretty general idea, general framework. You don't have to do this genetic or genomics.
I see. Yeah, that's great. Any any last minute questions? Anyone courageous? Well, if not, let's give our speaker one more virtual round of applause. And thank you so much for giving a virtual talk to our department and looking forward to seeing you. Well, I'll see you in person for sure, but for the rest of us, hopefully you get to meet Didong in person sometime in the near future. Thank you so much,
Great.
guys. See you
Thanks
next
for
week.
coming.